Hello, everyone. Thanks for your patience and giving me some time to complete chapter 3 with you. I think we're going to take advantage of these recordings if this one goes right. Maybe I can do this for chapters 4 and 5 as well. That way, we won't have to spend as much time on screen time, that is, on Saturdays, so that we can complete our chapters and keep us kind of moving along with the content, especially on days like a Saturday when we want to kind of get our days going. It would also be helpful if you could also review some lectures. Never know, you may have to miss a class or whatever that might be. So we'll look at this option if this goes well. Real quick, as we were progressing through our conversation about the cell structure last week, I said we would finish up with the cell cycle. And that's pretty much the remaining portion of the particular chapter that we're on. And so when we talk about the cell cycle, there's two types of cell division. The first is mitosis or meiotic cell division, and then there's meiosis. Meiosis occurs in sex cells, and that would be those cells that ultimately become a sperm or an ovum. And then there are your mitotic cells, which are the cells that your body cells, somatic cells is a better word to use other than body cells. That's what we're going to consider today. Those cells that ultimately become uh, parts of your body, your organs, organ tissue, and we're going to look at the actual cycle itself and the implications of the division and what happens as a result. So the first phase of the cell cycle, let me back up. The first phase of the cell cycle is interphase. And for a long time, it was thought that interphase was just a really short division or phase, but scientists have discovered interphase is actually about 24 hours long. So the average cell stays in interphase for a period of about 24 hours. It's a preparation phase, if you will, in which the cell prepares to be divided into two new cells. So there's a lot of prep work that has to happen. So you take interphase and you can divide it up into three individual subphases. The first is gap one, which is a rigorous growth and metabolism phase. The cell will permanently cease dividing at when you get to that point, or what they call like a G0, G naught, if you will. There's the synthesis phase, and this is when we replicate our DNA because we're going to have two copies of this original parent cell. So the two new daughter cells will need a copy of that DNA. And then there's G2, which is the preparation for division. This is essentially what it looks like on a big scale. If you'll notice, interphase is identified by the sort of turquoise color, and notice it's broken up into its three subphases. And then the remaining phases, which are prophase, metaphase, anaphase, telophase, and cytokinesis, make up the last remaining part of the cycle. Here we see on this side of the if you can see my arrow, there's a parent cell and it goes through the cycle, and then at the very end there are two new daughter cells where the arrow is closer to the darker blue there. So as I mentioned, we're going to go through a DNA replication phase, and it's really important that we go through this portion of the content because you have to understand that DNA replication is a very tedious, detailed process. And if you'll remember from the previous lecture, DNA is a double helix molecule. And essentially, we have to unzip it so that it can be used separately and replicated by RNA. Remember? So we're going to take the latter, that is DNA, and we're going to split it. And that's essentially what's happening in this image. An enzyme, DNA polymerase, is going to come through and unzip our DNA molecule here on the far left end of the image. And as it unzips it, it's going to make it longer, if you will, and make two new strands of DNA because you have to have an identical copy of the DNA for the two new daughter cells. After the replication is complete, the two DNA molecules are formed from the original. It's what's called a semi-conservative process, and you won't be tested on that, but essentially what it means is that it is an exact copy of the old strand of DNA, and there's no variations from it. It is identical, and that's why your cells, when we talk about somatic and body cells, that's why they all essentially are identical. When you think of the cells that make up the tissue in your lungs, those cells have to be the exact same. We're talking about body cells. So you have to make an exact copy of those cells so that the function of each cell can be carried out the exact same. So now we move from interphase, which you'll remember is broken up into the three subphases and is approximately 24 hours long, to the remaining processes, the remaining phases, should I say, of cell division and what we call the mitotic phases. Those are prophase, metaphase, anaphase, and telophase. And then a physical separation called cytokinesis. So let's take a quick look at those phases really quick. Prophase can be divided up into early prophase and then there's late prophase. The reason why it's divided up into early prophase and late prophase is because there's a process in which, and I know I sort of skipped over all of that, there's a process in which the cell membrane, or the nuclear envelope, should I say, begins to break down, and you'll begin to see the cell get really large. That process is our key marquees of this particular phase. So you know you're in early prophase when you begin to see these what they call centrioles move to opposite poles, but they're not quite there just yet. But the cell does get larger compared to what it was before because we've had to duplicate all of the organelles, we've had to duplicate the DNA, we've had to double in cytoplasm, and the chromosomes are starting to get much bigger and visible as well. So that's early prophase. But notice when you get to late prophase, the chromosomes have begun to sort of separate and and align themselves in sort of a organized fashion, but not as organized as they will be shortly. But also that the centrioles that are here, and there's one here that will be used to attach to the actual chromosomes, will reach out, the tentacles will reach out, grab them, and pull them apart. So prophase is kind of really just still more of a preparation phase, but the one thing for sure you're going to see is that the nuclear envelope will break down, and you won't see that DNA encased in the nucleus anymore. You don't even see the nucleus anymore as depicted by our images here. Metaphase, our centromeres of chromosomes are precisely aligned at the equatorial plate. So now our chromosomes are lined up right in the middle in what's called our metaphase plate, or sort of an imaginary line. And so here's our metaphase plate, and that's where the chromosomes are now lined up. Our centrioles are here and here, and they are ready to extend out what are called spindle fibers. And I think I called them tubules earlier, but they're spindle fibers. And they're going to reach out and grab each one will attach to a chromosome in preparation to pull it apart. Anaphase is the shortest of all phases, and that is essentially when the chromosomes begin to separate and pull apart and move towards the opposite end of the poles as the microtubules begin to, or the spindle fibers begin to pull apart. So you start to see anaphase, which is an indication there's a gap between the chromosomes right there in the middle, that they're actually moving away from the center, or what we call the metaphase plate. Telophase begins when chromosomes stop movement. They stop the movement that was in anaphase, and each set of chromosomes recoils, or uncoils, should I say, to form chromatin, which is what we really see when we look at the nucleus. The new nuclear envelopes form around each chromatin mass, and then the nucleoli appears, and then those spindles that I was describing disappear. Cytokinesis, it begins at the very late sort of anaphase, but actually completes during telophase. And that is essentially a splitting or a moving apart or a division of the two cells. And as you can see right down the middle of the two new cells is what's called a cleavage furrow. That's an indication that we have two new cells, and then this chromosome or the chromatin material begins to uncoil, condense, and then form our nucleoli, or that dark nuclear material that we saw in the very beginning. And that is essentially how cell division actually works. And those are the phases. If we were studying or looking at the sex cells, we would study meiosis, as I mentioned earlier. However, this is mitosis, so we don't necessarily have to go much further. There are a few other touch points that I'd like to kind of indicate or point out to you, not to sort of skip over, but when we talk about DNA replication and DNA synthesis, we have to take into consideration that there's our DNA, that the DNA that we are synthesizing or replicating inside of the actual nucleus prior to this is a lot more detailed, as I mentioned earlier. So remember, DNA is the master blueprint that holds the chromosome for protein synthesis. So DNA directs the order of amino acids in a polypeptide. A segment of DNA that holds the code of one polypeptide is referred to as a gene. So when you hear that word gene, it is a little bit more scientific than what we've kind of given it credit for being, or sort of given it the respect that it deserves. The code is determined by the specific order of nitrogenous bases, which is adenine, guanine, thymine, and cytosine. And a code consists of three sequential bases, so what we call a triplicate code. So you could have a G, and a C, and that particular sequence codes for an amino acid called proline. You could have GCC, which codes for arginine. Each triplet specifies the code for a particular amino acid. And then the genes are composed of axons and introns. Axons are a part of the gene that actually codes for an amino acid, and then the introns are non-coding segments that are sort of a interspersed amongst the exons. Sorry. So that's essentially the role of DNA. Now, how do we get from gene to protein? Like, that's actually a thing. You have DNA and you go from gene to protein. So you have the genes. Now, how do we get a protein? Well, RNA is the go-between molecule that links DNA to proteins. RNA consists of copies of the DNA code in a nucleus, and then it carries it into the cytoplasm to the actual ribosomes. All RNA is formed in the nucleus. And RNA, of course, differs from DNA as we discussed. Uracil is substituted for thymine in RNA. RNA has RNA has ribose instead of deoxyribose. And then there's three types of RNA. There's our messenger RNA, ribosomal RNA, and transfer RNA. A messenger RNA is really simple. All of the RNA is single-stranded, so that's a trick question or a trick note. All RNA is single-stranded, and what it does is it codes the DNA template and makes a copy of it in a process we call transcription. Messenger RNA contains the triplet codes from DNA. So DNA has its own code, but RNA will actually lay right on top of it and create a corresponding code for a particular amino acid. Ribosomal RNA is the structural component of ribosomes, the organelle where protein synthesis occurs. Along with tRNA, it helps to translate messages from messenger RNA to what's called polypeptide. Remember, polypeptide is a protein. Transfer RNA carries an amino acid, or is a carrier of an amino acid, has special areas that contain specific triplet codes, and there's also what's called an anticodon, which can also stop and start a particular amino acid from becoming a protein. So there's a chain of these codes, and then one can actually, what's called an anticode, can stop it at a point when it needs to stop. Anticodons of tRNA will complement base pair with codons of messenger RNA at the ribosome, adding in specific amino acids in growing the chain itself. And this process is called translation. So there's transcription, and then there's translation, and that is the process. So from gene to protein, we have to first transcribe our DNA, and that is using messenger RNA. And then from messenger RNA is decoded to assemble our polypeptides or our proteins in what we call translation. This is a graphic for you guys just to kind of peek at and check out. There's your DNA molecule at the very top. Then there's the transcription, which is the RNA processing it, and that's mRNA. Then as you go down, this is happening in the nucleus. Then there's transfer RNA, which brings it out. And then you begin the translation process. I'm going to skip this slide. This one. This is just a breakdown of the things I've just described. So quickly, I'll talk about the translation, and we'll probably land right here. Translation of protein synthesis is where the language of the nucleotide bases that I mentioned earlier, adenine, guanine, cytosine, and thymine, are translated. So just to look at that a little bit closer, remember our bases, adenine, uracil, cytosine, and guanine, um, are all the codes. And so these are the RNA codes. The DNA codes, you would have a T here. So we're actually essentially writing code based off RNA triplets. So these three will represent a triplet, and you'd have additional ones. And then it mentions the stop code. Now remember, there's 20 possible amino acids, but there's several codes that can code for one particular amino acid. Here's the chart of the genetic code. On my end, it's a little bit small, but what I'll tell you is that's for sure uracil, cytosine, adenine, and guanine. And then there's another set on this side, another set on this side. So this one represents the code on the left, this one represents the code in the middle, and this one represents the code at the end. And notice how some of them have 4 codes for one particular amino acid. Some of them have 2. Some have like 3 or 4. So there's a lot of codes. And all the letters are essentially the same. You know what I'm saying? Like not the same, but they're the same. They're just kind of in different orders. But notice here on this side, there's a quick chart to tell you what particular amino acid is being coded for. So you get the idea that these are the RNA codes. This is not DNA. This is what RNA would create from DNA to create the code. I'm skipping just a little bit because we've talked about these already. So to kind of go back to codes just a little bit, when we do have a length of code, a long length of code, like a sequence, if you will, that codes for a polypeptide. I mentioned earlier that there were stop codons, and here are the stop codons that would stop it. So if you saw 3 codes, another 3 codes, another 3 codes, now you have a polypeptide chain. As soon as this pulls up, it stops, it terminates the translation, and then you have the protein that you were coding for at the time, the final product essentially. This is sort of like a rendering of that, if you will. You should be able to see the lettering. I can't see it from my side, but it's actually reading the code here. And all this is happening in a ribosome. This is ribosomes that are found floating inside the cell. So in summary, um, The complementary base pair directs the transfer of genetic information in DNA to amino acid sequence of a protein. And the DNA triplets are coded by the messenger RNA codons, and those messenger RNA codons are base paired with transfer RNA anticodons to ensure the correct amino acid sequence. Remember, just don't forget DNA contains thymine, whereas RNA contains uracil, and then you'll be okay. And everything else is the same as it relates to this. So that's my 20-minute lecture. I would normally open up for questions from you, of course, at the end of class, but you guys are not here. So I will say, if you do have any questions or you have a comment, shoot me an email. We will have class this Saturday. We'll jump right into chapter 4 and wrap up chapter 5 in preparation for our exam, which has been rescheduled for October 19th. It's not this weekend, it's October 19th. That's all I have. You guys have been awesome, and I will see you on Saturday morning. Take care.